Thank you, everyone. I hope my voice doesn't go out. I've been recovering from a little bit of illness here, but my name is David Taylor. I'm from the Adesnik lab here at Berkeley, and today I'm going to be talking about some of the neuroanatomy underlying touch. Touch is part of the somatosensory system, which is responsible for letting us feel things like touch, pain, temperature, and giving us an innate feel or sense of how our body is positioned in space. And understanding how the somatosensory system works is an important role in neuroscience. Some of you may be familiar with the concept of the somatotopic map, which is the idea that points on your body are represented by points on the brain. Yes. Sorry. Okay. Does this go? Okay. So as you can see, the layout of the body on the cortex is more or less accurate. The face is all stuck together, the hand is next to the arm, but the proportions are completely wrong. Look at how different the lips and the hand are compared to the torso and the leg. They're completely wrong. This is strange, but it makes sense because I use my hands and my lips to discriminate texture and objects and shape. I use them as tools, so it makes sense that I would have that higher resolution in these parts of my body. Mice also have a somatotopic map, but it's prioritized a bit differently. Mice evolved to be nocturnal, and they evolved to live underground in subterranean environments in the dark. As a result, they have very poor eyesight. However, they're very good at navigating via touch. And they primarily use these long principal whiskers on their snout to navigate the world around them and perceive their environment. And the wake mice are constantly whisking these whiskers back and forth. And when the whisker comes into contact with an object, the signal is sent through the brainstem and the thalamus until it reaches a structure in the brain called the barocortex. And the barocortex is interesting because there's a relationship between the whiskers on the side of the snout and these barrel-shaped structures, hence the name of the barocortex, which is located in the cortex, the outermost layer of the brain. So there's a direct one-to-one relationship on the mouse's somatotopic map. And after the touch signal reaches the barocortex, there's some internal processing between the different layers of the cortex and between and within the barrel. And then the signal is distributed to other regions of the brain, regions involved with further sensory integration and processing, motor control and planning, and the thalamus, which I briefly mentioned before. The thalamus is kind of it distributes information to the cortex from all the sensory systems except for one. And I've been focusing on one part of the thalamus called the POM down here. And you notice that it's receiving projections from the barocortex, but I said that it also distributes information to the cortex. So a more accurate depiction might be like this, where the POM, part of the thalamus, is an intermediate between the whisker and the barrel. And then there's this kind of feedback loop between the thalamus and the barocortex. So let me back up just a bit. Previous research has basically told us all the different targets that the barocortex projects to. So every single one of these pink and red dots is a part of the brain that receives projections from this structure called the barocortex. However, this is about all we know. And what my project is trying to accomplish is to develop a more comprehensive survey, summary of these long-range projections. So we want to know how many neurons project to the POM. We want to know what layers project to the POM. The cortex is actually divided into roughly 6 layers, and they each have a different function. We want to know what kind of neurons are projecting to the POM. And finally, perhaps most importantly, we want to know, is there any overlap between neurons projecting to the POM and neurons projecting elsewhere, for example, the motor cortex. This is important because this would suggest that there's some common functionality in these neurons because they're projecting to 2 areas simultaneously. So finally, why is this important? I like to think of my project as developing a roadmap. A roadmap tells you how everything is connected. And so other neuroscientists can take that information, take that map, and do more complicated studies to determine why those areas are connected, not just how to connect them. So it's kind of just laying down a survey, a foundation that other people can build off of. So the methods I use involve 2 things, 2 components. It involves a gene and an enzyme that turns that gene on. And the enzyme is called Cre, and Cre will activate any gene that's flanked by a particular sequence. So since I'm trying to label neurons, what I do is I have the gene I'm using is a gene that expresses a fluorescent protein. So when Cre and this gene are combined, this Cre-dependent gene are combined, you get activation and the fluorescent protein is produced. And so the simplest setup looks like this. Real quick, neurons build up a voltage difference in the cell body until it reaches a certain point, and then action potential is released down the axon, where it synapses onto another neuron. So the flow of information is from left to right. And so what I do is I have genetically altered mice that have this Cre-dependent gene in every cell. So you can see it has the Cre-dependent gene in every cell. And then I inject a virus which contains the gene required to produce Cre into a region of my interest, for example, that's what I'm looking at now. And what happens is that this virus is taken up by the synapses and the cell bodies. So the cell bodies and the synapses that are in my region that I injected pick up the virus, it's transported backwards up to cells, and it activates the gene and fluorescent protein is produced, and the cell bodies and their axons are lit up. So an actual example of this is here. These are all neurons along the perimeter here. These are all neurons that project to my site of injection, which is the POM. So every one of these are all cell bodies, and they all have their axons projecting here, and they synapse onto neurons in the POM. So you have both these neurons that are projecting to the POM, and then these are all neurons in the POM that are projecting elsewhere. But I'm more interested in these ones in the cortex. And my data is in the form of brains, and I have a lot of it. So I'm going to just briefly take you through a small selection of what I produced over the summer, and kind of talk about what the applications might be of some of that. So just to orient you, I'm going to go through 3 sections, starting from the front of the brain to the back. And this was where I injected in the POM right there. So I'm going to start with the forwardmost section. And remember, in these sections, you're going to be looking towards the mouse's nose. Okay, so here's a section taken near the front of the brain. You can notice there's a brightly lit fluorescent area in the left hemisphere, and that is actually the motor cortex. Why might the motor cortex be projecting to the POM? Remember, all these neurons that you see will be projecting to the POM, which is where I injected my virus. So why might it be projecting there? Well, the motor cortex controls the whisking movement of the whiskers, of those long principal whiskers. And so it's thought that these neurons are actually telling the thalamus, the POM, what phase of the whisker is in. Is it all the way forward? Is it all the way back? Is it somewhere in between? And then using that, it can kind of determine where an object is located. If we move back, a little bit further, we can start to see part of the barrel cortex. And if you look very closely, you can kind of see a few of the barrels. I'll highlight them right there. You can kind of see each of the individual barrels. And you can also see these axon tracks, which are delivering axons to the POM and to the barrel cortex. So these reciprocal connections going back and forth. And if we move back one step further, this section actually shows the injection site at the POM. And it also shows the path of my needle tip. So you can actually track where exactly I insert the spikes. And again, you can see all these different axon tracks going from the POM to the barrel cortex and back. And you can also see the barrel cortex. And I want you to notice that most of the neurons are close to they're deep in the cortex. They're at the lower layers, layer 6 primarily. And I'm going to get back to this in just a second, but just want to keep that up. So over the summer, I accomplished a couple things. This was kind of an exploratory summer for me. The first thing I found was I found the correct coordinates. Even though there are brain atlases and literature to help you find the area that you're looking for, there's still a little bit of trial and error. And so through trial and error, I found regions that I'm fairly confident are the POM. And second, I found that layer 6 of the barrel cortex is the primary output to the POM. And this is unexpected because layer 5 is usually the layer that is the output layer of the cortex, but this was a little bit different. It was layer 6 instead. And this may be indicative of a kind of bottom-up, top-down dichotomy Bottom-up information is information that you get from your senses. So I mean, you get it from some receptors in your skin, your eyes, your ears, whatever. It has to come from an external source. Whereas top-down information is information that's actually generated by the brain. So there's kind of not really a struggle. There's kind of trade-off between bottom-up and top-down information, which one dominates. And in other systems, it's been observed that layer 6 is actually controlling how much the bottom-up information is running the game, or like the amplification of it. So it's kind of controlling how much impact it has. And so this layer 6 output from the neural cortex may suggest, maybe a sign that there is some regulation of how much top-down information has over the brain. And so as of now, I'm ready to enter the next step of my project, which is to use double injections. I didn't have time to go over that, but it basically allows me to more specifically label the neurons that I'm interested in. And I'm ready to start doing that now. At the same time, I'm going to start repeating this whole process for all those other brain structures I've talked about. So like the strident or the superior follicles, and obtain the same kind of data, and to eventually create like a summary of all these different regions that receive projection from the neural cortex. And finally, long term, the idea is you can transition from anatomical, where you label neurons, to functional, where you're actually turning those neurons on or off, and then observing how that affects behavior likewise. And I'd like to thank my PI, lab manager Lucy, and everyone else in the lab who all was kind enough to show up. Thank you so much. And I'd also like to thank Leah and Nathan for their hard work, and the Vibrissal Foundation for their financial support. Thank you, everyone.